In this video lecture, I will be talking on vaccination and immunoprophylaxis. Vaccination is a prevention of the disease by vaccine. The source of the word vaccine, it comes from the Latin word vacca, and it means cow. And this was on the honor of the British physician Edward Jenner, who pioneered the concept of vaccines and created the smallpox, the world's first vaccine. So, the term vaccine and vaccination are derived from varioli vaccinae, which means pustules of cow. Vaccines could be natural or artificial. Natural vaccine, it means that you don't seek service. You don't go to the hospital to get a vaccine or to get an injection containing a vaccine. You get it naturally. So I will give some examples of the natural vaccination. But however, they are first better to know that they are divided into natural passive vaccination, natural active vaccination. Natural passive vaccination, for example, during pregnancy, antibodies, they pass from the mother to the fetus. So the fetus will get antibodies, usually IgG antibodies. And also lactating mother. So milk from mother to the baby, also it provides the baby with IgA. So these antibodies will protect the fetus in the uterus and also during 6 months of life, the first 6 months of life. The natural active vaccination, also you don't seek service, but you get it naturally. For example, if someone get bitten by a mosquito, so the mosquito will inoculate sporozoites in his body. So this, it happens naturally, but however, following that, the body will see sporozoites and will upregulate immunity into an active way. So it is natural active vaccination. And now I am going to talk about artificial vaccination. Artificial vaccination, in this type of vaccination, usually individual seeks service. They go to hospital or dispensaries just to get vaccination. And it is also divided or subdivided into artificial passive vaccination and artificial active vaccination. Artificial passive vaccination, injection by ready-made antibodies. Here, the individual usually gets antibodies or ready-made antibodies. So these antibodies are not formed inside the body, but however they are raised maybe in animals. For example, protection against hepatitis A, or in the case of tetanus, the patient given antiserum containing antibodies against the toxins. This is anti-tetanus immunoglobulins. This is in a passive way. But however, artificial active vaccination, here the body is injected by either live attenuated vaccine or killed vaccine, or subunit vaccine, or recombinant vaccine, or toxoid vaccine, or by viral vector uh, vaccine. So now I am going to talk about artificial active vaccinations, different types of them. So I will start by just giving a short account on live attenuated vaccines. And in live attenuated vaccine, the pathogen get attenuated first and then delivered to the body. So the immune system starts to deal with it. Antigen presenting cells, as can be seen here, following injection of live attenuated vaccine, as can be seen here, the antigen presenting cell, usually macrophages and dendritic cells, they engulf the attenuated pathogen, as can be seen here in yellow. Inside they process it into peptides which bind to major histocompatibility class 2 at the rough endoplasmic reticulum. So peptides together with major histocompatibility will be expressed on the surface of antigen presenting cell, either dendritic cell or macrophages. Following that, usually T helper 1. T helper 1 cooperate with dendritic cell and with macrophages. They see peptides derived from live attenuated vaccine, and through TCR interact with the peptide, and through the CD8 interact with the major histocompatibility class 2 and get activated. I mean T helper 1 get activated and produce interleukin 2. And this interleukin 2 activates B cells, and also it activates T helper 2 cells. And B cell also, it is an antigen presenting cell. So they have the capacity, B cells, to internalize the attenuated pathogen by means of endocytosis, and also they present it to T helper 2. So the cooperation between B cell and T cell, this will result in production of some B cell growth cytokines, for example interleukin number 4. So B cell get activated and differentiate into memory B cell and plasma cell, which produce antibodies in the future infection upon encounter of the pathogen. So as I mentioned, that there is a plasma cell here, they produce antibody, and this is differentiated to memory cell, memory B cell, and remains in the body and recognize the pathogen if the body encounter it again, I mean future infection. Plasma cells, as can be seen here, produce antibodies, protect against pathogen, and if the pathogen affects the individual in the future, as can be seen here, the individual in the future. So these are some examples of live attenuated vaccine. For example, Bacillus calmette-guerin, which is attenuated Mycobacterium bovis and protects against human tuberculosis. Other examples are the measles, mumps, and rubella MMR vaccine, and the varicella chickenpox vaccine, and oral polio vaccine in form of Sabin vaccine in form of drops. They are given orally to children, and yellow fever vaccine. However, live attenuated vaccine may not be suitable for immunocompromised individuals. For example, individuals with autoimmune disorders, individuals with cancer, or individuals with some debilitating disease like tuberculosis, as these attenuated vaccine may regain their virulency and they may lead to the disease. So now I will talk on, or I will be talking on inactivated vaccines. These are not live vaccine, but however, one of the disadvantages of inactivated vaccines is that they do not give cellular immune response, and they stimulate only antibody immune response. In contrast to live attenuated vaccine, where the response is cellular and humoral immune responses. So the examples of inactivated vaccine are inactivated polio virus vaccine, whole cell pertussis whooping cough vaccine, and rabies vaccine, hepatitis A vaccine, tetanus, and diphtheria toxoid. So in this case, these vaccines do not deal with bacteria, but they deal with toxins expressed by some bacteria. And these actually toxins will be treated pharmacologically with means of formaldehyde to produce toxoid, which is not toxic to the human. But however, when the body upregulates against it, it will actually interact or work against different toxins by different organisms or bacteria. So now also I am going to talk about subunit vaccines, which is an active vaccine, and it is protein-based vaccine. Usually this vaccine targets protein on the surface of a virus, protein on the surface of a bacteria, or targets a secreted toxins. So subunit vaccines contain antigenic particles. It is not the whole microorganism, or recombinantly produced antigen or vaccine antigen in another organism. So examples for or of subunit vaccines are polysaccharide vaccines, for example, pneumococcal vaccine, conjugate vaccines. They have polysaccharide components stuck to a protein, for example, Haemophilus influenzae type B or pneumococcal vaccine. Now I'm going to talk about recombinant vaccine, and actually recombinant vaccine, it is a subunit vaccine, and in this type of vaccine, recombinant vaccine or production of vaccine recombinantly, this is DNA technology in which recombinant vaccine prepared by using another organism to produce the vaccine antigen. For example, grown in a culture with yeast culture of Saccharomyces cerevisiae, and this will lead to formation of a recombinant vaccine which harvests after that. And examples of recombinant vaccines are shingles vaccine, hepatitis B vaccine, a cellular pertussis vaccine. Toxoid vaccine, it is actually, these are toxins from some microorganisms, so they are harvested and inactivated by means of formaldehyde to produce a non-toxic toxoid antigen, or they produce toxoid vaccine. So the toxoid vaccine actually targets toxins by toxoid reactive neutralizing antibodies. Following injection of toxoid vaccine, the body will produce or upregulate antibodies against these toxoids, and this will fight the toxins. So an example of toxoid vaccines are tetanus vaccine and diphtheria vaccine. So now I'm going to talk about new technologies recently emerged, and I'm going to talk about viral vector-based vaccine. And this type of vaccine uses a viral vector, usually adenovirus, which is a non-harmful virus. So inside adenovirus is a genetic blueprint that encode the corona. For example, I will take a coronavirus as an example. Encode the coronavirus service protein. I mean the spikes or spike glyco protein. So when injected, the adenovirus interact with dendritic cell, as I mentioned before. This is similar, where the blueprint genetic material is transcribed into messenger RNA. But however, before that, they enter the nucleus in order to this step to be accomplished or fulfilled or achieved. So the messenger RNA, which get read by ribosomes as the cytoplasm or the cytosol of the human cell, and the coronavirus service protein is produced. So the cell will produce copies of the spikes or copies of the proteins of the spikes, which in turn presented on surface of dendritic cells, usually in association with major histocompatibility class 2. And at the secondary lymphoid tissue, usually lymph nodes, the dendritic cell presents these proteins to T helper, usually 2, and the T helper 2 help B cells to produce antibodies specific for the coronavirus service protein or spike glycoprotein. So examples of COVID-19 viral vector vaccine are Johnson & Johnson and AstraZeneca. Messenger RNA vaccine, it is more or less similar to this technique, but however, in this technique we use a virus, which is adenovirus, as a carrier for a genetic blueprint. But in case of messenger RNA, and as you know that messenger RNA is necessary for protein production, so we inject the blueprint directly, which is the messenger RNA of COVID-19. And this messenger RNA has the capacity to encode spike glycoproteins or service protein of the coronavirus. So the produced spikes will be either bind to major histocompatibility class 1, that means they will interact with T helper 1, and in this case T helper 1 will produce some materials and mediate cell mediated or cellular mediated immunity. So these memory cells will mediate immunity against the future infection. Or fragments of the spikes will be taken by the antigen presenting cell, which presents them to T helper, and the T helper will help B cell to produce antibodies that have the capacity to interfere with the binding of viral spikes to body cells in the future infection. So that is to say, messenger RNA vaccines stimulate both cellular and humoral immune responses, and the examples are Pfizer and Moderna vaccines. Thank you very much. Actually, I would like to thank Dr. Abir Abdel Munim for taking part in the preparation to this lecture. Thank you very much, Professor Ahmed Boulad.